Several common types of prescription drugs have mode of action that involve enzyme inhibition. Our group will discuss medication function in human body and how enzyme inhibitions can be used in positive manner in treating human disease.
Hello, for today's video, we are going to talk about prescription drugs that inhibit enzyme activity, namely ACE inhibitors, sulfa drugs, and penicillin. These three are common types of prescription drugs that have modes of action which involve enzyme inhibition. ACE inhibitors are used to treat high blood pressure conditions as well as several heart conditions. Sulfa drugs are a well-known family of antibiotics, and so are penicillin.
ACE inhibitors, are also known as the angiotensin-converting enzyme, is famous for antihypertensive medication and is good for congestive heart failure. Angiotensin is composed of an octapeptide hormone which is involved in blood pressure regulation. The goal of ACE inhibitor is to locate the angiotensinogen and convert it into an angiotensin, which can control blood pressure but may cause dry cough angioedema or hyperkalemia. Here we can see the structure of an ACE inhibitor.
A German bacteriologist named Gerhard Gaumack discovered the antibacterial activity of the compound sulfanilamide in the 1932, and this led to a whole family of sulfanilamide derivatives called sulfa drug, and this is the first known antibiotic in the medical field. Antibiotic is a substance that kills bacteria or inhibits their growth. Selective actions on bacteria that will not affect the normal metabolism of the host organism. Sulfanilamide inhibits bacterial growth because it is structurally similar to PABA. Sulfanilamide acts as a competitive inhibitor to enzymes in the biosynthetic pathway for converting PABA into folic acid in these bacteria. Here are the most common sulfa drugs: sulfanilamide, sulfacetamide, sulfasoxazole, sulfadiazine, sulfadimethoxy.
Penicillin, it is one of the most widely used antibiotics and was accidentally discovered by Alexander Fleming in 1928 while he was working with cultures of an infectious Staphylococcus bacterium. A decade later, the scientists Howard Florey and Ernst Chain isolated penicillin in pure form and proved its effectiveness as an antibiotic. Penicillins inhibit transpeptidase, an enzyme that catalyzes the formation of peptide crosslinks between polysaccharide strands in bacterial cell walls. These crosslinks strengthen cell walls. A strong cell wall is necessary to protect bacterium from lysis. By inhibiting transpeptidase, penicillin prevents the formation of a strong cell wall. Any osmotic or mechanical shock then causes lysis, killing the bacterium. Penicillin's unique action depends on two aspects of enzyme deactivation that have been discussed before: structural similarity to the enzyme's natural substrate and irreversible inhibition. Penicillin is highly specific in binding to the active site of transpeptidase. In this sense, it acts as a very selective competitive inhibitor. However, unlike a normal competitive inhibitor, once bound to the active site, the beta-lactam ring opens as the highly reactive amide bonds form a covalent bond to a critical serine residue required for formal catalytic action. The result is an irreversibly inhibited transpeptidase enzyme.
So that's it from our group. We hope that you have learned many things from this. Thank you for watching and God bless.